And here you can see some of the stuff we actually found. And here we have a medusa in the middle here, periphylla. And you can see we found some pretty big cod and also some capelin. And here some blue whiting. And you can see once we got further out, this is what we got. And I'll show you an image of that. So on the continental shelf and slope, we actually had some of these capelin, redfish, and Atlantic cod. And once you got into the lower continental slope, we got more into the squid, the Themisto, and also the plankton, the mesopelagic fishes we found some of, and also the jelly plankton. So you can see here, the gelatinous plankton are really important in the Arctic Ocean. Once we got out into the deep area, then you're talking about 4 kilometers to the bottom. And we are doing trawling and also plankton nets. And you can see here, we got no fish in the trawls, and we got arrowworms, large calendar copepods, deep water shrimp, and medusas, and ctenophores. Here are some of the jellies, and the jellies are actually the ones that are most abundant in the Arctic Ocean. And also these arrowworms, the Acronia hamata, these are pretty big arrowworms, and they are important. Here's the cruise track we had, and you can see this is based on hydroacoustics, EK80, and you can see that most of the catches we were registering with the hydroacoustics were along the coast of Svalbard. And once you get out into the Arctic Ocean, this is the way we went up. We went trailing behind French tourist icebreaker. So we could actually go from here to the North Pole in 2.5 days. And then we went east, and then we had this transect going back, and that was heavy with the Kronprins Haakon having to break all the ice for us moving. And here you can see again, here's the plankton. And you can see most of the plankton again are on the shelf and on the slope. And here's the capelin, yellow, and then cod in red. And once you get out in the Arctic Ocean, was very little. Then post-processing with the large-scale survey system, the LSSS system, Ole Arve took charge on that to get the values out of the hydroacoustics. Here's a recent paper that just came out from the Institute of Marine Research with Randi Ingvaldsen and company that very nicely sums up our current understanding of the Arctic Ocean. So in this case, you move from the Nansen Basin, which is closer to Svalbard, across the mountain range, the Gackel Ridge, into the Amundsen Basin, which is furthest away. And you can see that you get the fish on the slopes, and Atlantic water is here. Once you move out into the deep Arctic Ocean, you get ctenophores and jellies and some plankton. And once you move into the deep basins, then the jellies and the arrowworms are actually ruling the Arctic Ocean, and there's no fish. Thanks for listening.
Thanks, Haakon. We have time for a couple of questions, so I hope you in the audience can help me. Yeah.
I was just wondering if you did the fishing at different times of day and night to see if there's also like vertical traveling of the species?
There was some indication from the paper by the Germans from the Mosaic that there was some diel vertical migration in the plankton. But of course, at the time when we were up there in August, and then it's pretty much light all night and all day. So the diel vertical migration, when there's 24 hours of daylight, is not really a dominant feature. It's mainly when you get into the late autumn or early spring that you get the diurnal vertical migration. But in this case, you know, we put on a trawl at 400 meters and drag it up through 300 meters, and then you're certainly covering the whole diurnal vertical migration just by trawling. So that was not a problem. And the plankton net, we run from 1,000 meters up. So then we get everything in between.
Okay, we have a question a quick question up here.
Thanks. The plankton, you showed that there was high plankton abundance on the Svalbard shelf, then it went down, but then it also went up again at the pole, on the northern end. What features driving that, or what measurement was that plankton measurement?
This was based on the paper that just came out, and they admitted that it didn't do a proper plankton survey quantitatively for the whole transect that they had, and especially the one that showed very low abundance in between the coast and the shelf and then the Arctic Ocean. There was a lack of plankton survey in the middle there. So that was the explanation why it was low. It was just a technical thing.
Okay, thanks again, Håkon. I think we'll move on to the next speaker, who's Morwen, and maybe he tells some troubling news about how climate models function in the Arctic.
Yes, thank you. Thank you for that nice introduction. I'm here to tell you why climate models cannot project future Arctic ocean Atlantification. As many of you know, the Arctic is changing faster than anywhere else on the planet. The Arctic is outpacing the global average, warming about 2 to 3 times faster. And the implications of this are many. We see an extensive sea ice loss, approximately 40% of the summer sea ice extent, and about 2/3 of the sea ice volume has been lost. But we also see large changes in the Arctic Ocean marine environment. So in general, the Arctic Ocean is stratified by salinity. It's made up of a warm layer of saline Atlantic water at depth. And above that, we find a colder layer of fresher polar waters. And these two layers are separated by a very strong density gradient, which we call the halocline. Now, in recent decades, we've seen that a warmer and a stronger inflow of Atlantic water has changed this stratification. We also see, at the same time, changes happening to the surface layer due to an increased freshwater input from rivers, glacial melt, and sea ice melting. The changes in the Atlantic layer and in the surface layer, however, they have opposing effects on stratification, and therefore different manifestations in the different regions of the Arctic. So commonly we refer to this as the pacification of the western Arctic and the Atlantification of the eastern or Eurasian Arctic. So the pacification is mainly the effect related to increased inflow of Pacific waters, but also increased content of fresh meltwater, particularly in the Beaufort Gyre. And this is associated with a strengthening of the stratification in the Eurasian and Canadian basins. Whereas on the other side, on our side of the Arctic, the Eurasian basin, we see a warming inflow of Atlantic water, more Atlantic water, and this has been associated with a weakening of the stratification here and a weakened halocline. And this has a huge influence, of course, on the ecosystem with increased production and this northward transitioning, expansion of boreal species, and transitioning into an area which becomes more and more similar to the North Atlantic. Now, the big questions is, of course, what will happen in the future? Which of these mechanisms will dominate and where? So we have these two things happening at the same time that have opposing effects on stratification. Will the Arctic become more stratified due to increased freshwater, or less due to a more and warmer Atlantic water? Now, predicting the future is incredibly hard, but luckily we have some good tools to do this, and that's what we've been looking into. So we use climate models and combine these with future emission scenarios, socioeconomic pathways, and we use these models to then try to predict future change of the Arctic and also identify all these important feedback mechanisms in the very complicated Arctic climate system. The challenge, however, is that future projections always have some degree of uncertainty, just like any weather forecast has an uncertainty for a couple of days ahead. And the forecast improved depending both on the amount of data that we input into our models, but also the understanding of our processes. And I really like this figure here made by the Alfred Wegener Institute, which shows the spread or the uncertainty of future temperature productions as a function of latitude. We have the tropics in the middle. And as we see, we move rightward into the figure on the right-hand side in the Arctic. The uncertainty or the spread in the models is much larger than anywhere else on our planet. And that is, A, because we have very limited data from this region, particularly during winter, to feed our models with. But also there's very poor representation in these climate models of polar-specific processes. These climate models, they've gradually improved, and they're improving all the time. We've gone a long way from CMIP3 to now CMIP6 climate models. And what we've been working on in 2 new studies is to evaluate the newest versions, CMIP6 models, on how they perform in the Arctic. So we have 2 recent papers just came out, one focusing on these opposing trends in the future projections of Atlantification and Pacification. And we have another paper where we really focus more on the main state of these models in the Arctic and their biases and the causes for these biases. So just very shortly, unfortunately, the news isn't that good. We show in these papers that the CMIP6, the newest models, still struggle to accurately represent Arctic climate, the mean state, and the variability and the climate change. There's unfortunately larger biases in these models in the Arctic than anywhere else on the planet. The figure on the right-hand side shows some vertical profiles of mean ocean properties of salinity and temperature, and you see there's a huge spread among the models in how well they show the Arctic. Generally, the models get it too warm and too saline Atlantic water layer too deep, and generally the stratification is too weak, and they also struggle to get the main circulation patterns correctly. So this is a challenge if we want to use them to predict what's happening in the future. But of course, the main aim of our work has currently been to identify which mechanisms are responsible for these biases, which are the important processes that we need to work on to improve the models to get a more certain projection of the future. And a couple of the things we point to is that these models really struggle with the representation of shelf processes particularly, and also the connection between the surface and the deep ocean. Many of these models cannot get this ventilation correct, which is very, very important. And there's a lot of other things that the models still struggle with. But despite this, it's not only a doom and gloom. The models are not great, but they can still be useful. All models are wrong, we know that, but some are more wrong. But we can still use them to answer some questions. For example, all the models, they agree on a future strengthening of the stratification in the Western Arctic. There is some spread, as you can see in this figure right here, but all the models agree that due to increased freshwater, the Atlantification will not spread eastward. But on our side of the Arctic, in the Eurasian Basin, it's a whole different story. There's a complete divergence amongst the models in terms of future stratification. Some project future strengthening, some project a weakening. And this is a challenge because it can be either or according to this ensemble here. So overall, there is a need for improvement of all these models and their coupling particularly. We've now identified which building blocks we need to work on and which of the processes we need to improve in order to gain smaller uncertainty. But particularly what we also see that there's a huge demand for more observational data to constrain and to validate these models. And then we particularly need long time series, such as from moorings, so that we get the whole full seasonal cycle and winter data. If you use models in your studies as a researcher, some of the important take-home message here is, of course, to know the limitations of your models. There's a lot of data out there. It can be useful for answering some questions, but also they can't answer anything. So really be careful what the models can do and what they cannot. And also, since we have this spread and this divergence, be careful to not draw your conclusions on a single model system, because another model might show a complete different result. That being said, also using multi-model means is not a great idea, because if you have two opposites and you average them out, you actually show something or your results that's not represented by any model at all. So using a large ensemble of model is very, very useful. And with that, I would like to thank you for your attention.
Well, thanks, Morwen. You perfectly used up your time, so I can advise to hunt him down after the session and ask questions. Next up, we have Vivi. We will dive in a bit more deeper with some sperm whales.
All right, hi everyone. So I'm going to talk about the seasonal distribution of sperm whales in the high Arctic. So I'm gonna start off right away with some just basic information about the species itself, which is the sperm whale. It's the largest of the toothed whales, and it has a very cosmopolitan distribution. As you can see here on the map on the bottom right, their global distribution really ranges from pole to pole. Now, females and calves, they actually remain at low latitudes year-round, while mature males are the ones that migrate to the Arctic. But how far up north do they actually go? Well, we know that they are regular visitors to the Arctic, and sightings have been documented all the way from northern Norway to northern Svalbard. And very interestingly, in recent years, there's been a shift documented in the Svalbard sightings. But sperm whales are still limited from year-round access to the high Arctic, generally because of sea ice. And even though we know that sperm whales are regular visitors to the high Arctic, we actually don't know that much about the exact timing or the extent of their visits to this area, also because of challenges in data collection because of, again, sea ice, but also polar night. And actually these sightings are mainly from summer or early fall, because that's when these areas are more easily accessible. Well, one solution for year-round data collection, or a solution that allows year-round data collection, is actually passive acoustic monitoring. And this can, for example, involve deploying hydrophones on oceanographic moorings, which is the case in our study. And then these hydrophones can just stay there and record data practically year-round. And, well, it just so happens that sperm whales make an excellent candidate for passive acoustic monitoring or any kind of acoustic research, actually, because they're very vocal animals and vocalize most of the time. They produce clicks specifically for echolocation, but also use different types of clicks for communication. And previous studies estimated that these clicks can actually travel up to 80 kilometers in the water, which means that we can potentially hear them from quite far away. And this, of course, is very convenient for data collection with remote hydrophones. And let's talk a little bit more about the study itself that we did. And we had three main goals, and the first one was to actually find the sperm whale clicks in these hydrophone recordings, because the hydrophones, they of course record everything that's in the environment within their reach. So we first have to find the signals of interest, which in this case were sperm whale clicks. And then based on these detections, the goal was to create a baseline for spatial and temporal presence of sperm whales in the high Arctic, so around Svalbard, which was our study area. And then lastly, we wanted to do a little bit of a preliminary investigation of the possible drivers that could influence the found distribution. So acoustic data was recorded at 8 sites around Svalbard, as you can see here on the map, between 2012 and 2021. And in each recording period, the hydrophone was generally deployed in August, and then it would stay there throughout the winter until the next summer when the data was retrieved. And well, how did we detect sperm whale clicks or find these sperm whale clicks in the hydrophone recordings? That involved a two-part process, and the first one was an automated click detector, which essentially, in short, processed the files and tried to find click-like sounds, and then classified these click-like sounds as either non-sperm whale clicks or potential sperm whale clicks. And then these potential sperm whale clicks, or the files containing them, were assessed manually to confirm that they indeed were true sperm whale clicks and not something else. And this automated detector was incredibly useful because it decreased the manual workload significantly. We were actually able to process a total of 190,000 sound files, and only 5% of that had to be assessed manually. And let's get to the results, which are very exciting because sperm whales were actually detected at 6 out of the 8 locations around Svalbard, and 3 of these sites had multi-year presence, so every recording year at these sites, sperm whales were present. There were, however, very big differences in detection rates by site, and that's also illustrated by this huge figure here. So here, just the bottom x-axis is the full 10-year timeline, and then each row is a location, or each block. And if you look at the black bars, they represent the number of days with sperm whale detections per month, and you can see that the number and the height of these bars varies quite a bit. And what this figure also shows us is a certain seasonality in these detections. All detections in all locations occurred between May and January, so no detections at all at any locations occurred in February, March, or April. And then we went looking into some potential drivers of this that could explain this distribution, and the first one was bathymetric features, which is probably the most evident one, because if you just look at this map, you can see that the 3 locations with the most detections are all located in deeper areas at a shelf edge or a deeper trench. But then there's this one, which is kind of an odd one out, because clearly you can see that it's on a shelf edge, but there was 0 detections during 6 recording years. And this is where sea ice comes in as a negative influence. So as you can see on this map, which shows ice cover around Svalbard, on the western Fram Strait, it's very icy, so the red color is for very close drift ice. And then off northern and western Svalbard, it's pretty much open water. And this is just an example from a random day, but the situation is similar year-round. So the ice likely explains the absence of sperm whales at that one location. And this is also where we can see the influence of Atlantic water transported by the West Spitzbergen current. However, sperm whales were also absent a lot during open water periods, so there must be other factors that come into play. And this could be, for example, prey availability, or very likely it has an effect. And so we looked at the effect of zooplankton concentration, which had a positive effect on sperm whale occurrence, but we don't know the exact diet of sperm whales in Svalbard, so of course it's hard to investigate that directly. There is a potential prey though, which is the Borealisation armhook squid, and if you look at its distribution map here, you can see that the density is elevated at the locations where we found most sperm whales, so likely there is a link there. So then just to conclude and summarize, sperm whales were detected at most of the recording sites around Svalbard within almost year-round presence at some hotspots, and even the northernmost site at 81 degrees north had regular presence of sperm whales. And factors like bathymetry and prey density had a positive effect, whereas ice had a negative effect on sperm whale occurrence. And what this means for the future, well, the northward shift that already has been documented is likely to continue because more habitat opens up for sperm whales as sea ice declines. And now that we know a little bit more about the seasonality and the whereabouts of sperm whales around Svalbard, it's important to continue year-round monitoring, but also look more into the drivers so that we can then better understand and predict these future shifts. So this was my talk. Thank you for your attention.
Okay, thanks, Vivi. I think we can open for one quick question. No? Okay, thanks. And next up, we should have Julia, who's presenting on, we're up in the atmosphere right now, I suppose, so we're talking about birds. Yes. Please, go ahead.
Yeah, so today I'm going to talk about the effect of local weather on seabirds breeding in the Canadian high Arctic. So first, I just want to thank all of the people involved in this project. Long-term monitoring projects take a lot of people, a lot of time, and a lot of work, and this wouldn't be possible without them. So as we've talked about already, the Arctic is warming faster than anywhere else on the globe, which is causing large sea ice declines, big changes in extreme weather events, and resulting impacts on a lot of wildlife species. One group that's particularly susceptible to climate change is Arctic seabirds. Seabirds, climate change can alter their breeding habitats, their migration routes, and their stopover sites, as well as the distribution of their prey and their predators, and their breeding success, survival, and overall population trends. And in addition to these birds being vulnerable to changes, they're also really great indicators of these changes. These birds are at high trophic positions in the food web. They have central large foraging ranges around their breeding colonies. They are long-lived, so they can be monitored for long times, and they're also quite accessible at their breeding colonies in the summer. But all of these things that make them really great indicators also make them susceptible. So one of the most abundant gull species in the world, the black-legged kittiwake, is a really great indicator of the environment and has been used across the European and Canadian Arctic for decades. These birds are relatively long-lived. They have a circumpolar to mid-temperate distribution, and they're surface feeders, mainly feeding on fish like polar cod. But what's really unique about these birds is that they kind of operate at their energetic ceiling during breeding, which just means they're really sensitive to environmental changes and make good indicators. But like I said, this also makes them susceptible to threats. And these birds globally are considered vulnerable on the IUCN Red List for threatened species. But this differs a lot throughout the Arctic. In mainland Norway and Svalbard, these birds are considered endangered and threatened. Whereas in Iceland and Greenland, these birds are considered vulnerable, and the populations are decreasing throughout this region. In Russia, they haven't been formally assessed, and they haven't either in Canada, but we've seen, based on some population trends, that the birds seem to be increasing. And so we're trying to figure out what that may be and why. As climate change impacts continue to increase, it's really important to understand how extreme weather events and changes in ice will impact birds across their range. So, for example, in the European Arctic, studies have shown that strong winds can cause decreases in chick growth and survival. But we really don't know a lot about how weather impacts birds in the North American Arctic, like in Canada. And so that's kind of where my project comes in. So my first objective was to look at how local weather impacts the daily survival of eggs and chicks. And then also to look at how weather influences the seasonal differences in breeding success. And so our focus area is Prince Leopold Island in Nunavut, Canada. So this is a really productive marine area and is designated as a migratory bird sanctuary and important bird area in Canada. And it actually holds 16% of the Canadian population of black-legged kittiwakes. And what's great about this area is it's been intermittently monitored since the 1970s. So there's a really great long-term monitoring program there. But what's unique about Prince Leopold Island is it's yet to experience these large changes and fluctuations in sea ice and weather. So this acts as a really great way to start monitoring climate change impacts before they're severe. So we'll have a benchmark for future change. And so how did I do this? So we have daily breeding success metrics, egg loss and chick loss, which just means did an egg fall out of the nest and disappear, or did a chick die or fall out of the nest, for 2 years, 2012 and 2013. And then we also looked at weather. So we have a 3-day average on the day of the loss and the 2 days prior. And we looked at maximum wind speed, cumulative precipitation, and temperature to see how those may impact loss. In terms of the seasonal data, we have a few more years. So we look at the median hatch date for eggs across 13 years in between 1976 and 2014. And then we also have fledge success, which just means the bird grew up and was able to leave the nest for another 8 years. And we're looking at the same 3 weather metrics, but this time at a 1-month scale. So 1 month prior to hatching or fledging, how did that influence the birds? So first, into the results, I want to show the variability of weather on Prince Leopold Island. So this is a quick graph of what wind looks like. So the 2 years that are plotted, the 2 colors are 2012 and 2013, which is the days we look at daily egg and chick loss. But the range for all of the years is down at the bottom. So you can see that there's a lot of fluctuation day to day for wind. In terms of total precipitation, you can see that there's also a lot of differences day to day, but also in between years. And we see very similar things with temperature. There's a very big, like a broad change throughout the breeding season, but also in between years, there's a lot of differences. So how would this impact the birds? So these are the results of daily egg and chick loss. So this is just the proportion of eggs lost per day. And again, the colors are just the 2 years that we have. So overall, between 0 and 5% on average were lost per day for eggs, but we had a maximum of 16% loss in 2013. In terms of chicks, we see about 1 to 2% of chick loss per day, with a maximum of 20% right there. And so the question is weather causing this loss? No. So there was no relationship between daily egg loss or chick loss with wind, precipitation, or temperature. So now let's look and go back at the seasonal scale, where we have more years of data. So this is kind of the range of the median hatch date. So the years are on the bottom here, and you can see that the median hatch date varies a lot. Similarly, the proportion of chicks that have fledged in each year also varies a lot. So is this due to weather? No. Again, we found no relationship between the median hatch date and fledge success and these 3 weather variables. So we have a bunch of negative results. What does this really mean? Well, for one, unlike in Europe, kittiwakes don't seem to be impacted by these small changes in weather. But like I mentioned, we haven't seen those extremely strong changes in weather in the region around Prince Leopold Island. Here's a quick example. This is a graph of air temperature, and you can see that the Canadian Arctic is circled here in blue, the area around Prince Leopold Island, and it hasn't experienced as much warming as elsewhere. So in the short term, kittiwakes actually may fare well in the face of climate change. But as you've seen from the presentations earlier today, this isn't going to continue. So as climate change impacts continue to increase, this will be important to continue monitoring, as it will start to have an impact on these birds. So if not weather, what really could be driving breeding success? Many things. Maybe it's sea ice, maybe it's prey availability, and maybe it's predation. While we don't have any data on prey availability or predation at Prince Leopold Island, our next step will be to examine daily changes in both sea ice and sea surface temperature around the colony and see how that impacts daily egg and chick loss. And we recommend that future research does assess those prey availability and predation influences. So overall, the key takeaway here is that black-legged kittiwakes in the Canadian Arctic, which are really breeding at the high limit of their northern range, they don't seem that affected by small changes in weather now, but continued monitoring will be extremely important. Thank you.
Thanks for a clear and shotgun presentation, so you left plenty of time for questions. Anyone?
If you were going to be looking at predators as a potential new area to research, have you got any specific predators in mind?
Yeah, so on Prince Leopold Island, we have nesting glaucous gulls, and the glaucous gulls are really smart, and they've timed when their chicks hatch to be at the peak chick season for the rest of the birds on the island. So the glaucous gulls love taking black-legged kittiwake eggs or chicks, and so that's one of the predators we'd really like to look into.
Next question.
And I guess I'll take this from the other side. Prey has been shown to be important in fledging success, and by Tony and his group with other birds in some of the same areas and in the Hudson Bay. The stressors that you're using or the drivers that you're using are quite far from, I mean, unless the parents or the chicks are freezing to death, maybe there's something in between there that could be actually the factor that's driving that.
Yeah, that's one of the things we're hoping to see with sea ice. So black-legged kittiwakes in Arctic Canada primarily feed on ice-associated polar cod. And so ice should be theoretically a better predictor. But so we will see with our daily data if we see that, because at least that's a little bit more closely related to their prey, but we don't have daily observations of what they were eating, unfortunately.
Okay, Håkon, if you can be quick.
I was going to ask the one from below, but there are some surveys by DFO on fish in the Lancaster Sound, so you may want to dip into that and see what's happening to cod and capelin for that matter.
Yeah, that would be super great. We won't be able to do that on the daily level, but on that like large-scale seasonal level, that would be really awesome. The kittiwakes in the Canadian Arctic haven't seen that shift to more like Atlantification species like capelin and sandlance like kittiwakes have in Europe. They're still primarily only eating Arctic cod, so that's going to be the main influence. So it'd be interesting to see what the DFO surveys have to say. Thank you.
Okay, thanks. Thanks a lot. Next up, we're moving again to the opposite, an opposite place in the system. So Hedvig, next up, presenting more on crustaceans. Go ahead.
Okay, thank you. Hey, so thank you for all who are still here. It's also very much end of the day for me, so thank you for sticking. So in the next few minutes, I will present to you part of my PhD, which is dealing with the population expansion of boreal crustaceans in the high Arctic. And I guess I don't really need to introduce the concept of Atlantification in this session, but generally my work focuses on more of the biological aspects of the Atlantification and more specifically the population dynamics and the population expansion of species of boreal origin in the Arctic. So we actually 2 years ago published a paper on that, a review, so if you would like to check it out, then you are more than welcome. And within this study, we focus on 2 crustacean species, both of them which are intertidal. And basically, they have different dispersal potentials. And the different dispersal potential is a direct consequence of their different life cycles. So on the left, you can see Gammarus oceanicus, which is an amphipod crustacean. And what you need to know about them, that they brood both of the eggs and the juveniles under the marsupium, and the juveniles upon release are with very low mobility and thus very low dispersal potential. Sorry, I will just... Yeah. The other species that we studied is Samibalanus balanoides, which is the complete opposite, meaning that it has 2 types of planktonic larvae within its life cycle, both naupli and cyprid larvae, and thus it has a very high dispersal potential because these larvae can settle even several hundreds of kilometers from their release. In our dataset, we have both, and I forgot to mention that here we use DNA barcoding, which is a very simple methodology of sequencing mitochondrial cytochrome oxidase 1 gene and conducting population genetic analysis on this one marker. So in our dataset, we have both publicly available and newly generated CO1 sequences. And on these maps, you can see with the colors the regions, and the black dots are the sampling sites. So basically individual populations, which for some analysis are grouped into the marked regions. Very quickly about the methods. So we constructed very simple neighbor joining tree just to illustrate the genetic distances within species. Structure analysis to assess the hypothetical number of demographic groups. Then after that, we plotted the scaled genetic distances between populations, just to see what are the relations, the genetic relations between the sampling regions and sampling sites. And at the end, we did a very simple statistical test called mismatch distribution to test for the absence or the presence of current population expansion in the case of both species. So first, Gammarus oceanicus, which is, if you remember, the species with the low dispersal potential. So on the left, you can see a simple split tree, and what is very easily visible for the first look, that there is a big split between two lineages and a smaller one here in the second one. And our structure analysis supports this tree. Basically, we identified 3 hypothetical demographic groups, and one of them is placed in the Western Atlantic, together with the one site that we had from Arctic Canada for the species. This orange one is in the Eastern Atlantic, also Baltic Sea, White Sea, Barents Sea, and Norwegian Sea. And the third, which we call the Arctic group, is present in Greenland, Hudson Bay, and Svalbard. And the ancestry of the Iceland individuals can be related either to the second or to the third group. In the case of Semibalanus, the pattern is quite different. So if you look at the tree, it's a lot more messy, if you can call it like that. But basically, you cannot see like such a well-defined split as in the case of the previous species. Here you can see a branch which is genetically more distant from the rest of the dataset. But when you look at the structure plot, first of all, we only identified two groups, and both of these groups are present in all of our sampling regions. So after that, we took sampling sites, so populations as a unit, and we plotted their genetic distances between each other, scale genetic distances. And I know that it's a lot of colors and a lot of points, but basically what you need to see in the case of Gammarus oceanicus, you can immediately see the Western Atlantic group together with Arctic Canada, and here the Eastern group, and here is the Arctic, so Svalbard, the purple one, and with pink and red, Hudson Bay and Greenland. What is interesting here actually is Semibalanus, because even though on the previous slide you could see that with structure we identified 2 demographic units, but when we compare individual sampling sites, individual populations, they quite well group together geographically. So you can see this little cloud here is Svalbard, Western Atlantic, here is North Sea and the British Isles. So this is quite interesting. Lastly, we did the mismatch distribution, which is just a statistical test for the presence or the absence of population expansion. And here we have 3 main groups, so like Arctic, West Atlantic, and East Atlantic. And in the case of Gammarus, what we can see is there is a clear signal of population expansion in the Arctic, not so much, based on this mismatch distribution, not so much in the Western and Eastern Atlantic. And if we check the same areas for Semibalanus, our mismatch distribution doesn't show this clear signal of population expansion. So just to summarize and conclude, we managed to detect both demographically and geographically structured populations in the case of Gammarus oceanicus, while for Semibalanus balanoides, we did not see a clear geographic structure. This can be directly concluded from the, or directly be a result from their different dispersal potentials. Then a non-metric scaling of between population genetic distances recovered the geographic regionalization in the case of both species. And our mismatch distribution only showed population expansion for Gammarus oceanicus in the Arctic, while such conclusion from this analysis could not be drawn for the other species. And at the end, what is really important for me here to emphasize as a population genetist, that here we worked with one gene, which is a mitochondrial gene. And in the case of population genetic studies in marine environment, and especially in the case of species with such a huge genome, it's very important to look at these patterns on the whole genome scale, and this is something that I'm currently working on. So maybe, hopefully, next year I can come here with those results and show you that, because there are some very interesting patterns as well. And with this, thank you for your attention.
Oh, thanks a lot. We do have time for questions, so I hope I can challenge, yes, good Håkan, thank you.
Thanks for a very interesting presentation. Do you think Gammarus oceanicus are taking over the fjords in Svalbard and forcing, overtaking the other Gammarus species there, such as Gammarus setosus and various others?
Yeah, definitely. So there are already results on that and papers published on that, how the Gammarus oceanicus exactly is taking over the habitats of Gammarus setosus, and there is like a long-term monitoring for that. Basically, now Gammarus oceanicus has taken over all of the western coast of Spitsbergen, and setosus is completely just retracted to the eastern coast.
Okay, thanks again. I think we'll try to move on to the last presenter, who should be online. So, yeah, can you hear us? Raphael?
Oh, it's very strange to see yourself on the screen. Hi, everyone. You can hear me?
Yes. So,
yes, okay.
Please
So I'm
go
going to
ahead.
share my screen. Oh. Can you give me a visual signal that you can see everything? Or, yes, okay, perfect. Well, hi everyone, my name is Raphael. I'm a postdoc at UNIS on Svalbard as of 2 weeks ago. But the work that I'm presenting here today was part of my PhD that I did in Tromsø at the university there. And before I start, I'd like to acknowledge the funding sources that supported this work, as well as the wonderful co-authors. So as the presentation before mine summarized so wonderfully, the benthic organisms that live on the seafloor have different reproductive strategies. Some, like these amphipods, for instance, have a completely benthic development, while others, like this brittle star, will send planktonic larvae to drift with the currents, which will eventually settle back down to the seafloor and grow to become adults. And there's actually quite a few taxa that use this, this life strategy of drifting with the currents at the larval stage. The region I'll be focusing on today, as many others, is the Barents Sea shelf, just north of Norway and Russia, and around Svalbard here. In this region, we have warm water currents moving north from the Atlantic, and opposite that, cold water moving south from the Arctic Ocean. And these two major water masses will meet at the polar front. And so the question here is, can some of these planktonic larval stages follow these water masses moving north into the Arctic? In other words, what is the origin of the larvae that we find in the Barents Sea shelf? And to address that question, we're using two strategies. First, comparing the larval community inside the Barents Sea to the adults on the sea floor and see if they match. And here we're making the hypothesis that most larvae are local, but that some may be advected in from further south. And then coupling that with a particle tracking analysis, and here making this the hypothesis that some particles from along the Norwegian coast might be able to reach Svalbard and the southern Barents Sea within 3 months. So first, for the larval and adult comparison, we're once again focusing in this area here, just around Svalbard. And we have 3 major regions of interest, one northwest of Svalbard, and then 2 on the Barents Sea shelf. The region in green here represents an area north of a polar front, so largely influenced by cold Arctic waters, whereas the pink region below represents a region that's influenced by warm Atlantic water. And each of the black dots on this map represents a sampling location where we took a zooplankton net to collect the planktonic larvae. And then we put the entire content of that net into the blender, literally, to make a soup, and then sequence the DNA out of that soup to get a list of DNA sequences, which we can then match on online databases to get a taxonomic list of species that are inside the plankton. And here focusing specifically on the larvae of benthic invertebrates. So now that we have this list of larvae that were part of that sample, we can compare that to the adults on the seafloor. And there we compiled already existing data sets, which are luckily quite extensive in this region, and then coupled that with the GBIF records. So looking at the results here, at the top, we find the map with our 3 regions of interest for reference. And on this figure here, we see once again 1 bar for each of those 3 regions. And on the y-axis, we have the number of larval taxa. So the dark part of the bar represents the taxa that were found to be local. So in other words, there was a corresponding adult on the seafloor just below. And then the pale part of the bar represents the taxa that were non-local. So the adult was not found in the same general region. And if we focus on these non-local taxa, we find that the majority are made up of annelids, so the worms, and mollusks, especially nudibranchs. And I'll come back to nudibranchs here in a second. So the next question then that we're asking is, well, okay, so for these non-local taxa, where are the closest adults? And this is what is depicted here on this map, once again, of the Arctic Ocean. And we have the Barents Sea here. So we find that the majority of taxa, for the majority of these non-local larvae, the closest adult was located inside the Barents Sea shelf, around Svalbard, or along the northern Norwegian coast, so quite nearby. But for some taxa, the closest adult was located quite a bit further away. For example, these 3 nudibranch species, for which the closest adult was found further south along the coast of Norway. 3 more nudibranch species along the Russian Arctic coast. And then we're moving even further away with this bivalve, whose closest adult is in the Canadian Arctic archipelago. And then perhaps most extreme of all is this Nemertean, for which the closest adult is in the Sea of Japan, about 6,000 kilometers away in the straight line. So now we're coupling this comparison of adult and larvae with the particle tracking analysis. And for this exercise, we release the particle every 4 kilometers in the grid in the North Atlantic, and then let them drift for 3 months. And then asked which of these particles eventually drifted into one of our 3 areas of interest. And here's an example of the results from this exercise, where we see once again the map around. We've got Svalbard here towards the top and the northern coast of Norway at the bottom, with our 3 regions of interest. And each of these dots, colored dots on the map, represents the starting location or origin of a particle that eventually drifted into one of these 3 regions. And what we find here is that for the 2 regions inside the Barents Sea shelf, most particles came from quite close by, I would say quite regional origin, within the northern and southern Barents, or now north or south of the polar front respectively. for the most part, whereas the region northwest of Svalbard here had quite a wide range of origins, with some particles originated from as far south as around the Lofoten Islands along the coast of Norway. And that's a transit of more than 10 degrees of latitude within that 3-month period, which is really quite substantial. And that's because they've been following that Atlantic current moving northward. So to wrap up, we found that some boreal taxa with long-lived larvae do probably have the means of reaching the Arctic during their planktonic phase, but it's not because they're able to get to the Arctic that they're necessarily able to survive and grow new populations there, as they'll need favorable both physical and biological conditions inside the Arctic for that to happen. Which are likely not present at the moment, but with climate change and Atlantification, we could expect changes both in the transport of larvae, but perhaps most importantly, in their ability to survive and compete with Arctic species inside the Arctic. So if these new species did settle and did compete with local Arctic taxa, we might start seeing shifts in diversity and disruption in ecosystem functioning. So I suggest that we keep our eyes open for these little guys because they might be messengers for change in the Arctic. So with that, thanks for your attention. The results of this study were recently published in Marine Ecology Progress Series. So I invite you to go take a look if you'd like more details. But thank you.
Well, thanks a lot. Perfect in timing too, so thanks for that. Can open for a few questions.
Thank you for a great presentation. I'm just wondering, because you were using databases for the adults, what's the coverage like in the Russian Arctic? Because that one species that was the nearest one was all the way in Japan. It just makes me wonder what the coverage for the surveys were.
Yeah, so the surveys that we used were compilations from the Norwegian side only of the Barents Sea, so did not include the Russian side. But we complemented that with GBIF. GBIF is a global database of species distributions, which a lot of people, including a lot of Russian institutions, have been contributing to. So it's certainly not comprehensive, but it's the best we could get in this case. But I will say, especially for this Nemertian, but probably for a lot of other taxa, I do not think that the larvae would have come as far as the Sea of Japan into the Barents Sea. So I certainly suspect that the species is more broadly distributed than what we currently know, very much.
Any more questions for Raphael? No? Well, thanks a lot again. And I think this concludes our session. I would say that please join us again tomorrow. It's pretty late, 11, so I'm sure you managed to recover by then. So thanks to Jenny who arranged the timing. Yes, and I would like to give a hand to the presenters, since you were so good on keeping your time, which I was impressed about, in fact. So thank you.